learners, greetings of the day. Welcome to SWAYAM course on microbial technology, week 6, lecture 18. Myself, Devashish Sahu, currently working as research head at Biouniver Life Science Private Limited, Bangalore. I have more than 13 years of experience in the field of biotechnology and biology. Today, we shall learn about an important subject that is related to metabolic pathway. But before we shall proceed, let us know what were the learning outcome of previous class aerobic and anaerobic respiration. Aerobic respiration and anaerobic respiration both are related to respiration. So, respiration is a critical biochemical process through which cell convert nutrient into energy. There are two primary types of respiration, aerobic and anaerobic. Aerobic respiration requires oxygen and yield a high amount of ATP, while anaerobic respiration does not require oxygen and result in lower ATP production. Both processes are essential for different physiological conditions and organisms. So, aerobic respiration, as we all know, occurs in mitochondria of eukaryotic cells. It involves four main stages: glycolysis. This stage occurs in the cytoplasm and involve the breakdown of glucose into pyruvate, producing a net gain of 2 ATP and 2 NADH molecules. Pyruvate decarboxylation. Pyruvate is transported into the mitochondria where it converts into acetyl CoA, releasing carbon dioxide and generating NADH. Krebs cycle. Acetyl CoA enters into the Krebs cycle which produces 2 ATP, 6 NADH, 2 FADH2 per glucose molecule, while releasing CO2 as waste product. Fourth is electron transport chain and oxidative phosphorylation. So, NADH and FADH2 donates electron to the ETC in the inner mitochondrial membrane. The flow of electron generates a proton gradient that drives ATP synthesis, producing approximately 34 ATP molecule per glucose molecule utilized. So, in general, the overall equation of aerobic respiration is C6H12O6, that is glucose, plus 6 CO2 oxygen gives 6 CO2 plus 6 H2O plus ATP. That ATP is the energy cell of the microorganisms. Significance: aerobic respiration is highly efficient, yielding up to 38 ATP molecule per glucose molecule. It is essential for high energy demanding activities and is prominent in multicellular organisms including humans. Coming to anaerobic respiration, anaerobic respirations occur in cytoplasm and include glycolysis followed by fermentation. There are two main types of fermentation: lactic acid fermentation and alcoholic fermentation. Lactic acid fermentation, pyruvate from glycolysis is reduced to lactic acid by NADH, regenerating NAD+. This process occurs in muscles under strenuous activity and in certain bacteria. So, the overall equation is C6H12O6 gives 2 molecules of lactic acid plus 2 ATP molecules, that is the energy of the living organism. Then alcoholic fermentation, pyruvate is converted into ethanol and CO2 in yeast and some plant. So, overall the equation is C6H12O6, that is the glucose, gives rise to 2 molecules of ethyl alcohol or ethanol, C2H5OH plus 2 carbon dioxide plus 2 ATP molecules. The significance of anaerobic respiration is less efficient, producing only 2 ATP molecule for glucose molecule. However, it allows organism to survive and generate energy in oxygen depleted environment. It is crucial for certain bacteria, yeast and muscle cell during intense work. Aerobic and anaerobic respiration are vital biochemical processes that provide energy for cellular activities. Aerobic respiration, which its higher ATP yield support energy intensive activities and predominate in oxygen rich environment. But in contrast, anaerobic respiration enables survival and energy production in oxygen limited conditions, highlighting the adaptability of organism to varying environmental context. So, understanding this process is crucial for insight into cellular metabolism, physiology and adaptation. Now, let us start our session on Entner Duodruff pathway. So, the Entner Duodruff or ED pathway is a metabolic pathway for the catabolism of glucose and other carbohydrate, distinct from well known glycolytic pathway or PPP pathway or pentose phosphate pathway. It is named after its discoverers Michael Duodruff and Nathan Entner, who elucidated this pathway in the mid 20th century. The ED pathway plays a crucial role in the metabolism of many bacteria, especially gram-negative bacteria enables for ED pathway. Examples are E. coli, Zymomonas, Pseudomonas, Rhizobium, Azotobacter, Agrobacterium, etc. This ED pathway is also seen in very few gram-positive bacteria such as Enterococcus faecalis and in some archaea. The ED pathway's primary function is to convert glucose into pyruvate, facilitating the production of ATP, NADPH and NADH. These molecules are essential for cellular energy and biosynthetic reactions, despite sharing the ultimate goal of pyruvate production with glycolysis. The ED pathway achieves this through a distinct sequence of biochemical reactions, resulting in different stoichiometries of energy carriers and metabolic intermediates. So, let us go towards the discovery and history. The identification of ED pathway stemmed from studies on bacterial glucose metabolism. Researchers observed that some bacteria did not conform to the known glycolytic or pentose phosphate pathway for glucose metabolism. So, Entner and Duodruff work elucidated a novel series of reactions that accounted this observation, leading to formal characterization of this pathway. The mechanism. The ED pathway is a series of biochemical reactions that convert glucose to pyruvate and glyceraldehyde 3-phosphate, producing ATP, NADPH and NADH. So, here this is a detailed step-by-step explanation of the reactions involved. Step 1, phosphorylation of glucose. Here, one glucose molecule plus one ATP molecule gives glucose 6-phosphate plus ADP. Here, the enzyme that is responsible for the transfer of phosphate from ATP to glucose is hexokinase or glucokinase. The pathway begins with phosphorylation of glucose to form glucose 6-phosphate. This reaction is catalyzed by hexokinase or glucokinase and require one molecule of ATP, which is converted to ADP. Step number 2, oxidation to 6-phosphogluconolactone. The reaction is that glucose 6-phosphate plus NADP plus gives rise to 6-phosphogluconolactone plus NADPH plus H plus. The enzyme responsible here is glucose 6-phosphate dehydrogenase. So, here what is happening? Glucose 6-phosphate is oxidized to 6-phosphogluconolactone. This step is catalyzed by glucose 6-phosphate dehydrogenase and involve the reduction of NADP plus to NADPH. Now, coming to the step 3 of ED pathway, that involves hydrolysis to 6-phosphogluconate. So, the 6-phosphogluconolactone that is produced in step 2 in combination to water H2O gives 6-phosphogluconate. The enzyme that is responsible is 6-phosphogluconolactonase. The 6-phosphoglucolactone is hydrolyzed to 6-phosphogluconate by the enzyme 6-phosphogluconolactonase. So, this reaction involves the addition of water molecules. Now, coming to the step 4. Here what happens? Here dehydration to 2-keto 3-deoxy 6-phosphogluconate KDPG. Here the reaction occurs, 6-phosphogluconate gives rise to 2-keto 3-deoxy 6-phosphogluconate KDPG plus water molecule. The enzyme that is responsible is 6-phosphogluconate dehydrase, because here there is removal of water molecule. 6-phosphogluconate undergo a dehydration reaction to form 2-keto 3-deoxy 6-phosphogluconate KDPG. This step is catalyzed by 6-phosphogluconate dehydratase and release a water molecule. Then step number 5, cleavage to pyruvate and glyceraldehyde 3-phosphate. So, here KDPG dissociates into pyruvate plus glyceraldehyde 3-phosphate. The enzyme responsible here is KDPG aldolase. KDPG is cleaved into 2 molecules, pyruvate and glyceraldehyde 3-phosphate. The reaction is catalyzed by KDPG aldolase. The pyruvate can now enter into the citric acid cycle, where while the glyceraldehyde 3-phosphate can proceed through glycolysis for further metabolism. So, the overall reaction summary is that glucose plus NADPH plus ADP plus inorganic phosphate gives rise to pyruvate plus glyceraldehyde 3-phosphate plus NADPH plus H plus ATP. So, the complete pathway result in conversion of one glucose molecule into one molecule of pyruvate, one molecule of glyceraldehyde 3-phosphate, one molecule of NADPH, one molecule of ATP and one proton. The glyceraldehyde 3-phosphate can be further metabolized through glyceraldehyde to produce another pyruvate and additional ATPs. The key enzymes that are involved in ATP ED pathways are hexokinase, glucose 6-phosphate dehydrogenase, 6-phosphoglucolactonase, 6-phosphogluconate dehydratase, KDPG aldolase. So, the key points of EDP pathway are: the ED pathway starts with glucose, which is phosphorylated to glucose 6-phosphate. Glucose 6-phosphate is oxidized to 6-phosphoglucolactone while reducing NADP plus to NADPH. The lactone is hydrolyzed to 6-phosphogluconate. 6-phosphogluconate undergoes dehydration to form KDPG. KDPG is then cleaved to produce pyruvate and glyceraldehyde 3-phosphate. So, ultimately glyceraldehyde 3-phosphate is the end material. Energetics. Let us discuss about the energetics. The ED pathway produces 1 ATP, 1 NADPH and 1 NADH per glucose molecule. Pyruvate can enter into the citric cycle, while glyceraldehyde 3-phosphate can proceed through glycolysis to produce additional pyruvate and ATP. This pathway provides both energy, that is the ATP, and reducing power NADP plus NADPH, which are crucial for biosynthetic reaction and combating oxidative stress. Comparison with other metabolic pathway. So, now let us discuss how this differs with the other metabolic pathway. First, coming to glycolysis. First is the energy yield. In glycolysis, 2 ATP and 2 NADH per glucose molecule are formed, whereas in ED pathway only 1 ATP, 1 NADPH and 1 NADH molecule is formed. Talking about the intermediates, glycolysis involves a more complex series of intermediates, while ED pathways have a fewer steps. Enzymes, glycolysis uses a different set of enzymes, while no overlap with the ED pathway. Next, comparison with the pentose phosphate pathway. The pentose phosphate pathway have the primary function to generate NADPH and ribose 5-phosphate. The energy yield in the PPP pathway does not produce ATP directly, but generate NADPH similarly to EDP pathway. The PPP pathway shares some of the intermediates with glycolysis. However, the ED pathway shares some initial step of oxidative phase of the PP pathway, particularly the production of NADPH from glucose 6-phosphate. However, the overall outcome and intermediates are different. Regulations. The regulations of ED pathway is critical for ensuring metabolic efficiency and adaptation to environment conditions. This regulation occur at multiple levels, including gene expression, enzyme activity and availability of substrate and cofactors. Key regulatory mechanisms include transcriptional regulation. So, transcriptional regulation depends upon regulatory proteins and transcription factors. For example, activator proteins. Certain activator proteins can enhance the transcription of the gene encoding enzymes for the ED pathway. Example, Pseudomonas putida. The HEX-R gene product regulate the expression of the genes involved in the ED pathway. Repressor proteins. Repressor can inhibit the transcription of ED pathway genes when the pathway is not needed. For instance, the CRC protein in Pseudomonas species can repress the ED pathway in presence of preferred carbon sources like succinates. Operant structure. Transcription regulation. Operant structure. In some bacteria, genes encoding enzymes of the ED pathway are organized in operants. This means they are transcribed together as a single mRNA transcript, allowing coordinated regulation. The EDD-EDA operon in E. coli is an example, which includes gene encoding 6-phosphogluconate dehydratase EDD and KDPG aldolase EDA. Regulation of ED by global regulatory system. The presence of preferred carbon source like glucose can repress the expression of ED pathway genes through catabolite repression. This ensures that the bacteria uses the most energy efficient carbon source first. Two-component system. Some bacteria uses two-component system to sense environmental changes and regulate the expression of ED pathway genes accordingly. This involves a sensor kinase and a response regulator. Second, regulation of the ED pathway by enzyme regulation. First is allosteric regulation. In the allosteric regulation, there is a point called as feedback inhibition. Enzymes in the ED pathway can be regulated by feedback inhibition, where the accumulation of end products inhibit the activity of enzymes early in the pathway. For example, glucose 6-phosphate dehydrogenase can be inhibited by NADPH, ensuring that NADPH levels are balanced. The second allosteric modification is by covalent modification, by phosphorylation or dephosphorylation. Some enzymes may be regulated through phosphorylation or dephosphorylation, alternating the activity in response to cellular signals. This type of regulation is more common in eukaryotic cell, but can occur in prokaryotic cells as well. The third one is substrate concentration or substrate availability. So, substrate concentration, the availability of glucose 6-phosphate and other intermediate directly influences the flux through the ED pathway. High level of glucose can enhance the pathway's activity. Then transport system, glucose uptake. The efficiency of the glucose transport into the cells affect the rate at which the ED pathway operates. Transport systems such as phosphotransferase system in bacteria can regulate glucose activity, glucose uptake, and external and internal glucose concentration. Then metabolite channeling. So the regulation of ED can also be regulated by metabolic channel, for example by metabolite pools. The intercellular concentration of NADP plus, NADPH and other metabolite play a critical role in regulating the ED pathway. An adequate supply of NADP plus is necessary for the oxidation of glucose 6 phosphate, and sufficient NADPH is required for anabolic reactions. Coming to the second one, oxygen level, aerobic and anaerobic conditions, that comes under the environmental factor. The ED pathway can be more or less active depending on the availability of oxygen. Some bacteria might prefer the ED pathway under aerobic condition due to production of NADPH, which is needed for oxidative stress resistance. So another pathway is called stress response, oxidative stress. Under this condition of oxidative stress, the ED pathway can be upregulated to produce more NADPH, which help in detoxifying the reactive oxygen species ROS. So then coming to the physiological roles of ED pathway. The ED pathway is particularly prominent in certain gram-negative bacteria, as we have discussed, in Pseudomonas, in Zymomonas and Azotobacter. These bacteria often inhabit environment where the ED pathway provides a competitive advantages. First advantage is energy pathway. The pathway's efficiency in converting glucose to pyruvate with fewer intermediates and or ATP yield is advantageous in environment with abundant glucose. Another physiological role of ED pathway is metabolic flexibility. The ED pathway offers metabolic flexibility, allowing organism to adapt to different environment conditions. It also provides an alternative to the EMP pathway, classically called as glycolysis, and the PPP pathway. One of the most important physiological role of ED pathway is adaptation to oxidative stress. The ED pathway generates NADPH through the activity of glucose 6 phosphate dehydrogenase, which is important for anabolic reactions and oxidative stress response. The production of NADPH through ED pathway helps in combating oxidative stress by providing reducing power. This mechanism produces NADPH that necessary for the regeneration of reduced glutathione and other antioxidant molecules. Then central carbon metabolism. The ED pathway intermediates feeds into other metabolic pathway linking glycolysis, TCA cycle and other biosynthetic pathway. It also provides intermediate such as pyruvate, glyceraldehyde 3 phosphate, which are precursor of various biosynthetic pathways. Another significance is that ecological niche adaptation. The presence of ED pathway in certain bacteria is often linked to ecological niches such as soil bacteria, example are Pseudomonas species, and gut microbiota. It allows this organism to efficiently metabolize various carbon sources available in the environment. Then pathogenicity and symbiosis is another significant factor of ED pathway. In some pathogenic bacteria, the ED pathway is linked to virulence. Pseudomonas aeruginosa uses ED pathway during infection. In symbiotic relationship such as those involving nitrogen fixing bacteria, the ED pathway supports energy demand of nitrogen fixation. Then in archaea, the physiological role of ED pathway is that particularly these archaea strive in the extreme environment. So in some archaea, particularly those in extreme environment, utilizes the ED pathway. This pathway role in archaea is less well understood but is thought to be crucial for thermophilic and halophilic adaptations. Certain thermophilic and halophilic archaea rely on ED pathway for glucose metabolism. It has been observed that the ED pathway enzymes in these organisms are adapted to functional optimality at higher temperature and higher salt concentration. Thermophilic adaptation. The stability of ED pathway enzymes at high temperature support metabolic activity in thermophilic archaea. Metabolic flexibility. The pathway allows archaea to adapt to fluctuating nutrient availability in extreme environment. Other significance are ecological and environmental significance. The ED pathway ecological significance lies in the role in enabling microbes to thrive in diverse and often challenging environment. Key aspects are nutrient cycle. The pathway contributes to the cycle of carbon and other nutrient in the ecosystem. Microbial interaction. Microorganism utilizes the ED pathway can influence the composition and dynamics of microbial community. Some of the biotechnological applications are metabolic engineering. The ED pathway unique attribute make it valuable target for metabolic engineering. The applications may include biofuel production. Engineering these microorganisms to optimize the ED pathway can enhance biochemical or biofuel production from renewable feedstocks. Second one, bioproduct synthesis. The pathway can be harnessed for the production of chemical and pharmaceutical through engineered microbial strains. Now, coming to the conclusion, the ED pathway is a versatile metabolic route that provide bacteria and archaea with the ability to efficiently utilize hexose, generate energy and produce NADPH. Its role extends beyond simple energy production, contributing to metabolic flexibility, adaptation to environmental stress and the maintenance of cellular redox balance. Understanding the physiological role of the ED pathway can provide insight into the metabolic strategy of diverse microorganisms and their adaptation to various ecological niches. Let us discuss the topics that will be covered in the next chapter. In the next chapter or in the next lecture, we will be discussing about microbial photosynthesis. So, microbial photosynthesis is a vital process carried out by a diverse group of organisms that includes cyanobacteria, green sulfur bacteria, purple bacteria and certain algae. These microorganisms harness light energy to convert carbon dioxide and water into organic compound and oxygen, forming the basis of food chain and influencing global carbon cycle. Unlike plant photosynthesis, microbial photosynthesis exhibits remarkable diversity in mechanism and ecological adaptations, enabling microbes to strive a wide range of environment from fresh water to extreme habitats like hot spring and salt flats. By exploring the principle and variation of microbial photosynthesis, we gain a deeper understanding of its role in ecosystem, its evolutionary significances and its potential application in biotechnology such as biofuel production and environmental remediations. Microbial photosynthesis is a vital process carried out in diverse group of microorganisms include cyanobacteria, green sulfur bacteria, purple bacteria and certain algae. These microorganisms harness light energy to convert carbon dioxide and water into organic compound and oxygen, forming the basis of food chain and influencing global cycle. So, thank you.